You could say this soybean field in Queenstown is Bill Rhodes' happy place. On this sultry mid-July morning, Bill is using a pair of tweezers and a very steady hand to pollinate row after row of soybeans.
For me, it's almost like therapy because I can come out and I know every day I'm going to be out here opening buds and pollinating them.
Bill is Vice President of Soybean Research at Schillinger Genetics. He plays the role of honeybee in hopes he can improve a soybean's nutritional qualities.
The amount of varieties we have in the block here, we have a couple hundred varieties planted out here in all the different dates. So that's what my job is, to try to match up the best ones with the other best ones. And sometimes it works, sometimes it doesn't.
They're not genetically modified. The Queen Anne's County Research Farm specializes in traditional breeding, made more efficient with modern technology. But before these soybeans of tomorrow make it to the lab, they've got to survive in the field.
It's very tedious.
Bill's son, Michael, the soybean crossing manager, doesn't always share his father's zen approach to the job.
There's no other way to cross soybeans just because, they're self-pollinating.
Soybeans don't need bees or anything else to procreate. If the flower on the plant has bloomed, that means it's too late. It's already self-pollinated. Michael's job is to get to the bud before it opens and insert another plant's genetic material, or its pollen.
We'll peel back the petioles on the bud, and as we get that exposed, we can see the flower petals. And then we'll pull those out, and there'll be a pistil in there. That is what you want to put your pollen on.
Time is of the essence. After about 10 minutes in the heat and humidity, the pollen is no good. That's where the pollen runners come in.
We have him running pollen back and forth because, for two reasons, it makes it very efficient and a person can get more crosses done. And number two, I'm getting a little age on me and it makes me a little sore getting up and down all the time.
Even if Bill and Michael do everything right, the chances of making a viable cross still aren't great.
You usually want to be around like 300, like 30%, which we compare it to batting averages. So that's why everybody tries to be around 300.
To put things in perspective, each year they cross-pollinate 8,000 plants, but only end up with 2 or 3 marketable varieties. It's a lot of work, but when it pays off, it pays off.
There's a lot different genetics in this type of soybean than there is in this one. But if you cross these two, you may come up with something very unique out of it. You may not, but if you do, it's probably going to be genetically a very unique type of line.
The right kind of unique is high in protein, low in sugar, disease-resistant, and high-yielding. But it takes generations to be certain a variety has all those characteristics. That's where DNA analysis and this light cycler machine comes in.
We have DNA extracted from our seed. It goes through the machine, and it's not manipulating the DNA, it's just a machine that can actually tell you if that gene exists in that variety or that experimental line.
So based on the melt curve.
To put it simply, lab manager Dave Hulbert uses the light cycler machine to melt the DNA.
Let's suppose we're looking for resistance to a disease.
Dave can tell whether a plant inherited certain traits by looking at a given gene's melting point.
If we can knock out the lines that don't have the traits we're looking for early in the program, it makes us more efficient as we go down the road with getting varieties.
And it's still a long road before the new and improved soybeans hit the market.
These guys might share the same parents, but this one's flowering now, and this one hasn't even started. So there's that. There's maturity involved here.
Each potential variety is reproduced several times over, allowing research assistant Eric Wright to track and evaluate each one's physical characteristics, like leaf size and flower color. The finalists go through yield tests, and just 2 or 3 will end up growing in a field near you, eventually finding their way into food products or animal feed.
It's been very rewarding to work on what I think are traits that can improve the end use of soybeans in the long term.